Hi, I'm Stephanie Terezi from the laboratory of Laurent Brosset in the Department of Molecular Microbiology and Immunology at Brown University. Today we will show you a procedure for the generation of KLRG1 tetramers. We use this procedure in our laboratory to study KLRG1 coherent interactions. Let's get started.
First, inoculate 4 500 milliliter flasks of TB buffer containing appropriate antibiotics with bacteria expressing the KLRG1 plasmid construct and grow at 37 degrees Celsius with shaking. When the OD reaches 0.6 angstroms at 600 nanometers, induce expression via the G7 promoter with the addition of 0.4 molar IPTG and incubate the culture for an additional 4 hours shaking at 37 degrees Celsius. Following the 4-hour incubation, transfer the bacteria to centrifuge tubes and spin the cultures down for 20 minutes at 3000 RPM and 4 degrees Celsius. If necessary, pellets can be frozen at -80 degrees Celsius at this point for use at a later time. Resuspend the harvested cells in 30 milliliters of resuspension buffer. Next, pool bacterial resuspensions up to a 60 milliliter volume in a polypropylene beaker. If necessary, bring the total volume up to 60 milliliters with extra TE buffer, pH 8, and stir the mixture at half speed on a hot plate stirrer. While the pooled resuspension is stirring, add the following solutions dropwise to the mixture: lysozyme, magnesium chloride, DNase 1, Triton X100, and finally DTT. Transfer the solution to ultracentrifuge tubes. Using a Fisher dismembrator with ultrasonic converter at 30% amplitude, sonicate the solution on ice for 1.5 minutes and centrifuge the lysate at 5000 RPMs for 10 minutes at 4 degrees Celsius. Decant the supernatant and add 15 milliliters of wash buffer. Repeat sonication as before until the pellet is completely resuspended and repeat centrifugation. Repeat the washing, sonicating, and pelleting steps 5 times. Finally, resuspend the pellet using wash buffer that does not contain Triton X100, spin down once more, and resuspend in 4 milliliters of TE buffer. These are your inclusion bodies. Quantify your inclusion bodies by wet weight. The inclusion body slurry can be kept at 4 degrees Celsius in TE buffer stocks of 30 to 100 mg per milliliter concentrations. With the inclusion bodies ready, you can now prepare for refolding. Begin the refolding procedure by preparing a fresh 1 liter bottle of refolding buffer and stir at low speed at 4 degrees Celsius to chill. Next, you will prepare 7 injections of the inclusion bodies for injection into the refolding buffer. First, melt the stock of inclusion body slurry in a microcentrifuge tube containing a final volume of 1.4 milliliters in 7 molar guanidine hydrochloride plus 10 millimolar beta-mercaptoethanol in a water bath at 37 degrees Celsius for 30 to 40 minutes, vortexing every 5 minutes. You will know the melting is complete when the slurry becomes clear. Centrifuge the melted mixture at max speed for 30 minutes in a microcentrifuge at 4 degrees Celsius. Then, being careful not to collect any of the small blackish pellet, transfer the supernatant to a 15 milliliter centrifuge tube. Adjust the volume to 7 milliliters with injection buffer. To inject, first increase the stirring speed of the folding mixture to high. Then add 1 milliliter of diluted inclusion bodies, drop by drop, with 2 seconds between each drop. Only add 1 milliliter of diluted inclusion bodies at a time, with an hour in between each injection. In between injections, return stir speed to low. Continue stirring at low speed at 4 degrees Celsius overnight. The proteins will then be ready for concentration and purification. To begin concentrating the refolded KLRG1 tetramers, first pre-filter the reaction using a 0.22 micrometer filter. Then concentrate the reaction from 1 liter to about 10 milliliters using a millipore Amicon stirred cell and a 10 kilodalton nominal molecular weight limit ultrafiltration membrane according to manufacturer's instructions. Then, using a millipore 10 kilodalton molecular weight cutoff ultrafiltration membrane, Concentrate further to less than or equal to 2 milliliters. Have your chromatography system set up according to manufacturer's instructions. We use the GE Healthcare Acta Fast Protein Liquid Chromatography System at 4 degrees Celsius. Using a high-resolution column, purify the monomer form of KLRG1 in 20 millimolar heaps and 150 millimolar sodium chloride by size exclusion chromatography. The volume injected should be less than 2% of the total gel volume of the column. Again, using a millipore 10 kilodalton molecular weight cutoff ultrafiltration membrane, concentrate further to a final volume of 1 milliliter. Biotenylate the KLRG1 monomer according to manufacturer's instructions. Eliminate the free biotin by purifying as before using size exclusion chromatography. To achieve tetramerization, mix the KLRG1 monomers with a 4-fold molar excess of phycoerythrin conjugated streptavidin. Tetramers are now available for labeling receptor ligands. This graph shows a positive result of separation of refolded KLRG1 proteins using size exclusion chromatography. From left to right, the peaks illustrate the multimer, dimer, and monomer forms of KLRG1. The peak on the far right represents the buffer exchange. Though refolding conditions have to be tested empirically for each protein, other C-type lectin receptors should be able to be tetramerized using a similar protocol, thus enabling the potential identification of other ligands.
We've just shown you how to generate the KLRG1 Tetramer. When doing this procedure, it's important to remember to check the induction levels and to strictly adhere to the refolding conditions. So that's it. Thanks for watching and good luck with your experiments.